This video will take you through the stages of meiosis, discuss the similarities and differences between mitosis and meiosis, and describe how meiosis relates to Mendelian genetics. The word meiosis was derived from the Greek maion, to lessen, and was aptly given because meiosis is the process through which a diploid cell reduces its genetic and chromosomal content by half to produce a haploid cell. As described in the previous video, mitosis produces two diploid daughter cells whose genetic material is identical to that of the parent cell. Meiosis, in contrast, produces four haploid gametes for sexual reproduction. As shown here, each haploid cell now has only one copy of the long chromosome and one of the short. Mendel had no concept of the molecular mechanisms that underlie his observations, but the events of meiosis can explain both Mendel's law of independent assortment and the law of independent segregation. Mendel's law of independent assortment states that two different genes randomly assort their alleles during the formation of gametes, while the law of independent segregation states that two copies of a gene separate from each other during gamete formation. Now let's get back to the mechanism. Mitosis and meiosis share a very similar beginning. For both processes, a cell first passes through the first three stages of the cell cycle, G1, S, and G2. But instead of continuing the cell cycle, the chromosomes that were duplicated during S phase then undergo two successive divisions, meiosis I and meiosis II. The cells, or gametes produced by meiosis, can go on to fertilization, if very, very lucky. The stages of meiosis I and II are the same as mitosis and include prophase, prometaphase, metaphase, anaphase, and telophase. First, here's what a cell might look like at the end of G2 after DNA synthesis. This should look familiar. I've drawn 6 pairs of sister chromatids, long, medium, and short pairs in red, maternal, and blue, paternal. Like in mitosis, the genetic content of the cell is 2N and the chromosomal content is 4C. Here are 2 homologous chromosomes. During prophase I of meiosis I, many of the same events happen that occur during mitosis, with some important differences. First, the chromosomes condense, similar to mitosis. But in meiosis, pairs of homologous chromosomes align with each other. This is a key difference from mitosis. How do the homologous chromosomes pair up? In fact, this phenomenon is not completely understood, but is generally attributed to the similar DNA sequences found in homologous chromosomes. Once the pairs of homologous chromosomes are connected as a single unit, they are known as a tetrad or a bivalent. Recall that a pair of sister chromatids is also called a dyad, while a single chromatid is known as a monad. Although we will not discuss it now, it is important to note that crossing over also occurs during prophase I. Similar to mitosis, in prophase I of meiosis, the nuclear membrane begins to degrade and the spindle apparatus begins to form. In prometaphase I, the spindle apparatus, or meiotic spindle, forms completely. Note, I'm not going to draw most of the polar microtubules, but they are present. In this stage, each kinetochore microtubule attaches to one dyad, meaning that each pair of sister chromatids is only connected to one spindle pole. Moreover, the orientation that the maternal and paternal dyads are bound by the kinetochore microtubules is random. For ease of illustration, most drawings depict the sister chromatids in the tetrad side by side. However, when it is shown as a stacked pair, it is easier to see how the sister chromatids attach to a single spindle pole of the meiotic spindle. This is distinct from mitosis, where each chromatid, or monad, is attached to a kinetochore microtubule. During metaphase I, the sister chromatids align at the metaphase plate due to the opposing tension of the kinetochore microtubules, but the direction they align in is random, so they could look like this, or like this. Let's stick with this arrangement I have drawn here. With 23 pairs of homologous chromosomes in humans, that's 2 to the nth power, or 2 to the 23rd power, or over 8 million possible arrangements. So which of Mendel's laws does this explain? If you were thinking Mendel's law of independent assortment, you are correct. In anaphase I, the bonds between the tetrad break and the dyads, or pairs of sister chromatids, are pulled apart. In telophase 1, the chromatids decondense, the nuclear membrane reforms, and cytokinesis splits the cell into 2 cells, each with 3 pairs of sister chromatids. The genetic content of the cell is now 1N because the homologous chromosomes have been separated, so there is only one allele for a given gene, barring crossing over. The chromosome content of the cell has also been halved and is now 2C. In a human cell, there would be 23 pairs of sister chromatids at this stage. Now, how does the result of meiosis 1 relate to Mendel's laws? Well, at this stage, the maternal and paternal alleles of any particular gene were separated into different cells, explaining the law of independent segregation. Therefore, these resulting cells can also be described as haploid. Meiosis 2 comes next and is very similar to mitosis, except that it begins with haploid cells. In prophase 2, the chromosomes recondense, the meiotic spindle begins to reform, and the nuclear membrane begins to break apart. In prometaphase, each chromatid in the pair of sister chromatids connects to a spindle pole, like in mitosis, but different from meiosis 1. In metaphase 2, the pairs of sister chromatids align along the metaphase plate. And in anaphase 2, the connection between sister chromatids is degraded, and the dyad is pulled apart into single chromatids. Finally, in telophase 2, the nuclear membrane reforms, and cytokinesis generates 4 haploid cells from 2 haploid cells. The genetic content is still 1N. As shown here, each cell now has 3 chromosomes, and the chromosome content is now 1C. The haploid cells produced by meiosis are called gametes. Over the course of meiosis, 4 haploid cells were produced from 1 diploid cell that went through 1 round of DNA synthesis, followed by 2 rounds of division. Meiosis comes full circle with fertilization. The fusion of 2 gametes, such as an egg and a sperm, generates a diploid zygote with double the genetic and chromosomal content of the gametes. For example, a human zygote will have 46 chromosomes total and is ready for mitosis to begin. Now it's time to consider the outcome of meiosis. Recall that each gamete is unique and that chromosome assortment alone generates over 8 million possible combinations. When 2 parental gametes combine, there are even more possibilities. Ultimately, meiosis generates variation in the next generation, increasing the odds that some offspring will reproduce and that the genetic material will be passed on through successive generations.